Hello to our friends joining us via recording. We are doing our second review session for exam number 3. Friendly reminder that this exam is open now and it closes Tuesday night, tomorrow night at 11:59 p.m. Make sure that you are giving yourself time to complete it by that time so that you get a score spit out as soon as it's done and you don't have to wait for Dr. Allis to go in and give you a score for it. So in our group work activity, before we started our class here together, we reviewed a lot of stuff from lesson number 9 about muscle tissue. So one of the things we talked about in relation to muscle tissue was some of the things that can go wrong with the process of muscle contraction. So when we talked about the process of muscle contraction, let me get some terminology up here for us. Remember that big picture. When we talked about the process of muscle contraction, we called it excitation, contraction, coupling. Excitation, contraction, coupling. This was the idea that I change my membrane charge, that's the excitation part, to make my muscle contract. That's the contraction part here. When we're talking about each of these different problems we can have with muscle contraction, they actually impact different parts of excitation, contraction, coupling, giving us problems. So let's start here with Botox. When we talk about Botox, big picture, what happens to a person's muscle when they're exposed to Botox? So biggest picture, how might we describe what Botox does? Yeah, I love it. So we are paralyzed. There's no contraction, right? So no muscle contraction. Our muscles are paralyzed. We can't do that. Yeah, so our extension to that, as Kaylin is mentioning, the reason why we don't do any muscle contraction is because Botox is an issue with us not sending messages to do muscle contraction. When we're talking about the message that neurons send to a muscle to tell them to contract, which of these things on my list right here is the message that a neuron sends? Yeah, exactly. So this is my message. Let me put a little box around it. Acetylcholine, ACH. That's my message that muscles receive telling them to contract. The reason in Botox that we have no muscle contraction is because we have no acetylcholine. So to get more specific with Botox, I'll type the whole name actually, no acetylcholine, ACH, is released. So big picture, my muscle can't contract. Well, why can't my muscle contract? Because I'm not releasing any acetylcholine. Here's where we get specific about the actual way that Botox works. The reason I'm not spitting out any acetylcholine is, remember, botulinum toxin, which is what Botox stands for, this toxin goes inside a motor neuron and it prevents it from being able to spit out acetylcholine. So no acetylcholine is released because Botox, I'm going to say Botoxin just because it sounds better. Botoxin is inside the neuron. Botulinum toxin, Botoxin, it goes inside the neuron. Remember, it kind of chops up the anchor or the ropes that we would have used to pull that acetylcholine bubble out of the cell. Yeah, so that's Fanshin's mentioning the specific name of those rope proteins that I use to pull the acetylcholine out of the cell. They're called the Snare Proteins. Exactly. So the Snare Proteins are some of my proteins that help me to spit out acetylcholine. Botulinum toxin goes in there, chops up those proteins. Now I'm not able to spit out acetylcholine anymore. So big picture, when someone is exposed to Botox, their muscles don't contract at all. They don't contract at all because they never get the message that they should be contracting. And the reason they don't get that message is because the neuron doesn't spit it out. This is what happens in Botox. We summarized it. Here's my summary, because we did a comparison in class, right? When we talk about Botox, this is an issue with not enough acetylcholine. Not enough acetylcholine. At the end of the day, if you don't have enough of this message, you can't contract. This is the message that makes us contract. Let's go over here to the next thing we talked about. We talked about sarin gas. Just like we did big picture here with Botox, what's big picture here with sarin gas? What's going on with muscle contraction and sarin gas? Yeah, exactly. So Kira mentioned we've got non-stop muscle contractions. With Botox, we had no muscle contractions. When we're talking about sarin gas, we just keep contracting and contracting and contracting. Yeah, so a couple of us have mentioned in the chat, sarin gas is a compound that doesn't mess with acetylcholine itself. It has nothing to do with acetylcholine like Botox does. Sarin gas comes down and messes with this guy right here, ACHE. Let me type its full name for us here. Acetylcholine esterase. Acetylcholine esterase. Sarin gas binds to acetylcholine esterase and prevents it from doing its job. So acetylcholine esterase is blocked by sarin gas. It's blocked by sarin gas because sarin gas likes to go into that protein and hold on to it and grab it tightly. When we talk about acetylcholine esterase, what's the normal job of this protein? In the process of muscle contraction, we've got to have it because it helps me to do something normally. Exactly, I like it. This is so I'm going to put normal job is to chop up ACH, to break down acetylcholine. Because remember, when a neuron spits out acetylcholine, that causes muscle contraction. But it should only cause muscle contraction once. We don't want to keep contracting and contracting and contracting for the same message that we sent 5 minutes ago. When we talk about being exposed to sarin gas, this is actually an issue not of not having enough acetylcholine. Now we have too much acetylcholine. So too much acetylcholine. We're summarizing biggest picture. What's going on with sarin gas? I have too much acetylcholine. The reason I have too much is because I'm not doing my normal job. I'm not chopping up the acetylcholine when I'm done hearing it. I just keep hearing it over and over and over again. So primarily when we talk about sarin gas, what I want you to think of as the main problem is acetylcholine esterase. We can't chop anymore because this enzyme is deactivated. And because it's deactivated, anything that we released in the past, we just keep hearing it over and over again. Yeah, so Tien's totally right. The amount of acetylcholine that I'm spitting out, the number of messages to contract, that's totally normal. My motor neuron is acting just fine. It's doing its normal job, except we're not breaking down those messages like we should be. So it looks like we have too much acetylcholine because we're never taking out the trash. It just keeps building up and building up. With sarin gas, we've got kind of a hoarder situation going on, if you will. So any acetylcholine that was released in the last 5 minutes, I'm going to keep hearing it. Just like a hoarder will keep things for years, right? So in sarin gas, we aren't taking out the trash. We're hoarding everything. Whereas in Botox, we're just not spitting out anything. There's no messages, period. Botox and sarin gas. When we think back to this process called excitation, contraction, coupling, which is the big picture way a muscle contracts, Botox and sarin gas both are issues with excitation. They're both issues with the way that a neuron talks to a muscle. Either, in the case of Botox, we don't talk at all, or in the case of sarin gas, we keep hearing the same message over and over again. Both of these are problems with excitation, with the message getting our muscle cell ready to contract. When we talk about rigor mortis, now we're talking about the contraction side of things. So we got our signal, that stuff's working just fine, but now we've got an issue with contraction. When we talk about rigor mortis, in easy words, what is rigor mortis? What is rigor mortis to a normal person? Yeah, exactly. This is what happens when your body gets completely stiff after you died. So body stiffening after death. That's my big picture. What's wrong? My body is stiffening. It's stuck, if you will, after death. This has to do with the thing, like Fanshin mentioned, called the Crossbridge Cycle. So the Crossbridge Cycle is the specific way that I go through the process of muscle contraction. Remember that process has 4 steps, so we do need to review those steps for the exam. We start with Crossbridge Formation, where myosin and actin get attached to each other. Then we've got the power stroke, where we actually move the things past each other. Then we've got crossbridge detachment, when actin and myosin let go of each other. And then we have the cocking of the myosin head, or the reactivation of myosin to get ready to do it again. All of those things are what happen in contraction, in the contraction part of excitation, contraction, coupling. When we talk about the body getting stiff when you've died, that's because we're not able to do one of those steps in the Crossbridge cycle. Do we remember from our studying? With rigor mortis, what can we not do? Yeah, exactly. Several of us chiming in. When we have rigor mortis, when the body is stiff, muscles cannot do detachment. The stage of the Crossbridge cycle where myosin and actin let go of each other. So what normally happens in crossbridge detachment is myosin and actin let go of each other. Part of that is to get us ready to contract again. Part of that is just because it keeps our muscles looser. Crossbridge detachment is what should happen. It does not happen in rigor mortis because there's something involved in this detachment stage that we don't have when we've died. What do we not have when we've died? Yeah, exactly. So one of my basically my biggest picture issue when we're talking about rigor mortis is I have no ATP. No ATP. This is the chemical that helps me to do crossbridge detachment. ATP comes along, attaches to myosin, and myosin will let go of actin. Well, if I have no ATP, I'm never going to let go. Just like one of my groups was mentioning, and we can see it here in the chat. Kaylin liked to give the analogy here when we're talking about what's wrong in rigor mortis is a Romeo and Juliet situation here. So Romeo and Juliet, my proteins here, actin and myosin, they just won't let go of each other, even if it causes them to die. So we won't let go. Our muscles are super stiff. That's rigor mortis. We are star-crossed lover proteins that will never let go because those proteins don't have ATP. So when we're thinking about rigor mortis, that's going to be an issue with actin and myosin. They are stuck together. They won't let go, star-crossed lovers, but also an issue with ATP because I don't make any ATP to help me detach them from each other. What questions do we still have, or thoughts do we still have about these disorders? Or give me a thumbs up if we feel okay about them. Oh wow, Kira gave me a star. She's feeling great. Good. Kaylin says she's feeling a little better. Awesome. Yeah, the more we can put these things in big picture terms, the easier it is for us to understand. So I'm glad to hear that's helping us. All right, well then let's talk about this ATP stuff right here. When we are talking about muscle tissue, we name our muscle tissues based on the way they make their ATP and the way that they use their ATP. So we have 3 types of skeletal muscle tissue in the body: fast glycolytic, fast oxidative, and slow oxidative. Let's start, before we hit our phrases over here, let's start with these words in their groups to help us remember what kinds of information their names tell us. So let's start with the second set of words in their names. When we're talking about glycolytic or oxidative, how might you describe, what kind of information does this tell me about a muscle tissue? Glycolytic or oxidative, what does that tell me about them? Yeah, perfect. So Kira kind of summarized, and Kaylin did kind of as well. Biggest picture when we're talking about these two, glycolytic or oxidative is how they make ATP. Big picture, how they do it. So there's 2 ways they do it, and several of you mentioned it in the chat for me. One way that we can make our ATP is we can use sugar. So my type that is called glycolytic is my kind that they're going to make their ATP using sugar. Specifically, the kind of sugar that they store, that's called glycogen, is the special sugar that skeletal muscle tissues store. So if you are a type of glycolytic muscle tissue, you used sugar, stored sugar, to build your ATP. If you are an oxidative kind of muscle tissue, you definitely use oxygen to help you make your ATP. What else could we say about an oxidative type? We use oxygen, and what else could we say? Yeah, so the mitochondria. Yeah, so we use mitochondria with that oxygen, that special thing called the electron transport chain. We use those things to make our ATP. So when you look at a type of muscle tissue, every time you look at it, glycolytic, that tells you right away, this is one that uses sugar to make its ATP. Oxidative, that tells you a whole lot of things, right? It tells you I've got to have oxygen to make my ATP. It tells you I use mitochondria to make my ATP. All of that information just in this last word of its name. And then we've got this first word up here, either fast or slow. In general terms, what might you say that fast or slow word tells us? What are some of the kinds of things we can get based on fast or slow? What does that tell us about them? I like it. So we've got both of the things, the kind of information we can get from these. Big picture, it tells us how quickly ATP is broken. Big picture, how fast you break down ATP. So if you are a fast type of muscle tissue, unsurprisingly, you break down ATP quickly. If you are a slow type of muscle tissue, unsurprisingly, you break down ATP slowly. But the speed that you break down ATP helps us to know whether you're making a strong contraction or a weak contraction. Help me out. Is it fast or slow types of muscle tissue that make strong contractions? Which one makes a strong contraction? Yeah, exactly. If you are a fast type of muscle tissue, you chop up ATP really quickly. And if you chop up ATP really quickly, that means you get that energy really fast. So a fast type of skeletal muscle tissue is going to make a really strong contraction. A slow type of muscle tissue is going to make a weaker contraction. It's still going to generate some energy, but it's not going to be very strong when it does its contraction. The corollary to that, though, has to do with the fact that we're breaking down ATP, right? If I chop up my ATP, or the example we gave with our lumberjacks was if I chop up my wood really slowly, it's going to take me a longer time to get through it. I might be able to be chopping for longer, or if I'm burning that wood, if I burn it slowly, it's going to last longer. Whereas with a fast type of muscle tissue, I'm going to chop up all that ATP really quickly, and I might run out if I don't have a lot of it. So a fast ATPase that uses ATP really quickly, and it helps us make really strong contractions. A slow type of muscle tissue has a slow ATPase, so it's going to use the ATP more slowly, but it's also going to make a muscle contraction that's not that strong. This is the information, either how quickly we break up the ATP or how we make that ATP. All of that information is hidden in the name of these types of muscle tissue. So as long as you know what these words mean in their name, you're going to be able to apply that to these statements here about our types of muscle tissue. So let's go through and look at these types of muscle tissue and remind ourselves. We're going to start here with our first statement that asks us about running out of ATP really quickly. Which type of muscle tissue, do we remember from our groups, would run out of ATP really quickly? Yeah, exactly. This is going to be my fast glycolytic muscle tissue. Fast glycolytic. Now, keep this in mind. When we talk about running out of ATP, there's two things that have to happen for me to run out of ATP. Thing number one that has to happen is I have to chop it really fast. For me to run out of ATP, first thing that's got to happen is I've got to be chopping fast. Because if I'm chopping slow, I'm never going to run out. But the second thing that has to happen for me to run out of ATP is I need to have a limited amount of ATP. In other words, I need to have not an indefinite supply. I need to not be really, really good at making ATP. Because when we talk about the mitochondria, when we talk about that thing called the electron transport chain, remember that we said that takes a little bit of time to get started, but once it gets started, it goes nuts and makes all the ATP. If I'm making all kinds of ATP, it doesn't matter if I'm chopping it up really quickly, I'm never going to run out. Yeah, with our lumberjack analogy, remember that we said that our oxidative types that use the electron transport chain, that lumberjack is chopping up the forest. He's never going to run out of wood. But if I just have a pile of wood, like I do with glycolytic proteins, if I just have a pile, I'm going to run out. Eventually, I'm going to chop my way through it. So when we talk about running out of ATP, I need to chop up any ATP I have really quickly, and I need to not be making a crazy amount of ATP. Based on these two things, we know that the answer to this question is fast glycolytic. Glycolytic going along with part 2, fast going along with part 1. See how we can use the name of these types of muscle tissue to help us predict what's going to happen with it. Let's look at our next one here. When we talk about carb loading, which type of muscle tissue did we say would benefit from carb loading? Who's going to benefit from carb loading? Yeah, exactly. This is another one that would be beneficial to fast glycolytic muscle tissues. We don't care so much about fast as much as we care about the glycolytic part of its name. When we talk about carb loading, carbohydrates are sugars. So sometimes they recommend that you eat a bunch of sugar to get ready for a sprint, for example. So you're loading up your muscles with extra glycogen. We're storing extra sugar. The type of muscle tissue that stores sugar and uses sugar is fast glycolytic muscle tissue. Emphasis on the glycolytic part, because that's the ones that care about sugar. Remember, my oxidative ones, they don't care about sugar because they're using that electron transport chain to make their energy. They're not using glycogen. They're not using sugar. They're using the electron transport chain. Our next statement asks us about myoglobin. When we talk about myoglobin, what is the function of myoglobin? What's the function of myoglobin? Yeah, Kira's right. This is all about storing or transporting oxygen. Transporting oxygen, myoglobin. Remember, it's like the little brother of hemoglobin. Hemoglobin in red blood cells, myoglobin in muscle cells. When we're wanting to figure out the type of muscle tissue that would have a lot of myoglobin, meaning something that transports oxygen, who's going to need a lot of oxygen? Not a trick question, I promise. Yeah, exactly. This is going to be, the ones that have a lot of myoglobin are going to be my fast oxidative muscle tissue and my slow oxidative muscle tissue. With these two types of muscle tissue here, the emphasis is on oxidative. Oxidative. That looks just like oxygen. If you're using oxygen in your mitochondria to make ATP, you are going to have a lot of myoglobin because that's what brings oxygen to your muscle cells. So I've got a lot of myoglobin if I'm oxidative. If I changed this statement and said, who doesn't have a lot of myoglobin? So basically I'd be asking, who doesn't need oxygen? That's going to be my glycolytic friends, right? They don't use oxygen, which is to their benefit. You can not have a ton of oxygen and still contract fast glycolytic muscle tissue. But remember, like we said up here, these ones are going to run out of ATP really fast because they don't use oxygen to make their ATP. They're stuck with the amount of sugar they have stored to make their ATP. So lots of myoglobin in my oxidative types of muscle tissue because they use that oxygen that myoglobin transports to make their energy. All right, let's look down at our last statement here. We're talking about a type of skeletal muscle that helps us to do repeated contractions, and it's something that takes a lot of energy, or we're making strong contractions. Here's where we can take hints from the statement and from what those words mean in their name to help us predict the name. So let's start here. When we talk about a lot of energy, this means we're doing a strong contraction. A strong contraction. What's the word that we used in the name of these muscles to tell us that they do strong contractions? What did we say meant strong? Yeah, exactly. Fast. The word in the name of these muscle tissues, if we are a fast muscle tissue, we do a strong contraction. Absolutely. Notice that our statement talks about repeated muscle contractions. This means I need to make a lot of ATP. I need to not run out of ATP. What word in my muscle type names means I'm going to make a lot of ATP and I'm not going to run out? Yeah, exactly. That word oxidative means that I'm going to make a lot of ATP. If I'm doing a repeated muscle contraction, meaning I'm not just doing it once, I'm doing it over and over again, that tells me we're talking about a type of oxidative muscle tissue. When it's a strong contraction, that tells me I need to have a fast type of muscle tissue. So based on what we figured out with the names, we're talking about fast oxidative skeletal muscle tissue. That's going to help us do things over and over again that require a lot of energy. I know at least one of my groups was talking, what was the activity that you had in your packet that I told you that fast oxidative muscle tissue is really good for? What's this really good for? Yeah, exactly. The example I gave you all with tennis. So doing those really strong bursts of energy to send that ball over the net, but you're not just doing it once, you're doing it over and over and over again. So fast oxidative muscle tissue. Here's my wink, wink, nudge, nudge for you. When you are taking the exam, see how we went through with these phrases and we looked for the kind of information that the question told me. So for example, right here we talked about how carb loading meant that we're doing sugar. This is a key for me, meaning sugar. Remember that when we're talking about our types of muscle tissue, glycolytic means sugar. Or when we talk about myoglobin that transports oxygen, remember that our oxidative types of muscle tissue use oxygen. Hey, I could have changed this phrase. Maybe instead of saying myoglobin, I could say that thing called the electron transport chain. That's the name of the part of the mitochondria where I make ATP inside of it. Mitochondria use oxygen that myoglobin transports. They use that oxygen in the electron transport chain to make ATP. So when we're talking mitochondria, when we're talking oxygen, we mentioned this thing, the electron transport chain, any of those things are tip-offs for you that we're talking about oxidative muscle tissue. And then when we talk about those contractions, it's all about whether we're doing strong or weak, maybe how quickly we're running out of ATP. There are always little context clues in your questions and in your answers to help you figure out what we're looking for. So use the names of these types of muscle tissue to help you out with phrases like this, and to help you out with everyone's favorite, critical thinking. By the way, how did we feel about this question? We'll talk through it, but I'm just curious. Thumbs up, thumbs down. Yeah, Kaylin wasn't a fan. Kier liked it. Kier found a trick. Okay. Well, let's talk through it and see if we can get to a place where it's not quite so frightening to us. So, like I mentioned, these are the kinds of questions that we sometimes see on the exam, right? So let's work on this one together to train us for our exam. This question starts by saying niclosamide is a medication known as a mitochondrial uncoupler. Now, I know one of my groups was like, what does this mean? Hey, guess what? It doesn't matter. I told you in our next sentence here, I know it's a distraction. Next sentence tells us what a mitochondrial uncoupler does. So a mitochondrial uncoupler makes it harder for mitochondria to make energy. So if you study in, I don't know, like a biology for majors class, the way that the mitochondria make energy is by there being a difference in the concentration of protons inside and outside. What a mitochondrial uncoupler does is it's like, psych, we're going to make it the same inside and outside, and then it's hard for the mitochondria to work. That doesn't matter to you, the way that a mitochondrial uncoupler works. What does matter to us, the only thing out of all this stuff that matters to us, is this right here. We're talking about mitochondria. We know that this medication makes it harder for mitochondria to make their energy. So we go down to the question we're actually answering. My actual question is, using what we know about the types of muscle tissue, so fast and slow, oxidative, glycolytic, and the way this works, which the way it works is it makes mitochondria have a hard time working. We need to predict who could be affected by this medication. So for my group that was freaking out about this a little bit, check this out. All we care about is this statement right here. This medication makes it hard for mitochondria to work. If we want to know who's going to be affected, which kinds of skeletal muscle tissues will be affected, we just need to know who's got mitochondria. All that matters for us, we don't care what it does to the mitochondria, we just care that it makes it harder for them to work. So if you don't have mitochondria, you don't care. You can take this medication all day and no big deal. But for some of my types of muscle tissue, like several of us are saying in the chat here, specifically the fast oxidative muscle tissue and the slow oxidative muscle tissue, remember that oxidative means I use the mitochondria to make my ATP. If you give me a medication that tells those mitochondria, you're not going to work so great, I'm going to be very strongly affected because that's my primary way of making ATP. Now, meanwhile, we've got those fast glycolytic muscle fibers over here, and they're like, mitochondria? Meh, don't care. These guys are not going to be affected. These guys are just plugging along, breaking up their glucose and doing muscle contraction. So this type of muscle tissue would not be affected by niclosamide because it doesn't use mitochondria to make its ATP. I promise you will see questions like this on the exam that look really scary, and there's distracting words in here that we don't know what they mean. But if you look past those words, or the way I like to tell students is, if you don't let them steal your head, don't lose your mind when you read this and you're like, oh my gosh, what's a mitochondrial uncoupler? I promise you don't need to know that. You just need to know, hey, look, mitochondria. hey, I know that I've got types of muscle tissue that use mitochondria. If this is going to make it harder for mitochondria to work, that's going to affect me because I have mitochondria. So with a question like this and any of the critical thinking questions on the exam, I promise you, hidden in those things that make you feel really nervous, that you don't like reading about words you can't understand or you can't pronounce, like niclosamide, you don't have to know, you don't need to know anything about that word. You can like cross it off and it can be anything. We don't care about what it is. We can say compound X. Compound X. Compound X is a medicine. That's all we care. We don't need to know how it works in particular. We just need to know this, that it makes it harder for mitochondria to do their job. If I know that, I can answer this question. So don't get caught up on the distractors. Critical thinking questions, I promise you, you've got the skills, you've got the knowledge to do it. I know you can do it. Just take a deep breath, look for any clues that you can cling to, and ignore the junk that you don't know about. I promise you'll make it through. How do we feel about this question now? Now that we've talked about how to tackle it. Or let me rephrase too. Are there any other particular questions that we have related to it? I know I've got several thumbs up, which is perfect. Okay. Well, I'm glad to hear we're feeling a lot better about it. I will post this video after class today. So if you want to go back and look at this question without all of my scribbles on it, right? To keep training your mind for what critical thinking questions look like. I know we had some of those type questions in Friday's session as well. So for those of you that ask a lot, how do I prepare for critical thinking questions? Go back and look at this one again. Go back to the beginning of each of those videos from our previous classes. I like to try to throw in some critical thinking questions, not always, but at least some of the time I throw in some critical thinking there. So check that out. All right, so the one topic that someone had mentioned was Botox. We did already talk about that. Do we want to talk more about Botox, or do we feel okay about Botox now that we worked through it on that first group work slide? Okay, so Fanshin says she's feeling good about that. Okay. Well, I need y'all to come up with some more topics. While you're thinking, I want to show you something really cool. Teacher nerd moment here. Let me get my screen to share here. This is something that I just recently learned about that is so fascinating to me. So this is something called the Connectome. If you do a Google search for the Connectome, this is showing you, remember that picture that I showed you when we were talking about the white matter of the brain, and I showed you that brain that was kind of chopped up? This is imaging studies that they've done to see the way that the brain is arranged. So I mentioned this briefly in class last week, that different personality types have different ways that they connect part of their brains to each other. These are some of the images of the way that different parts of the brain are connected to each other. Yeah, so Kira asked to relate it to our material. Yeah, these are the tracks in your brain. So they're just color-coded and they look so beautiful. I just love looking at this. So seeing those different functional areas, the way that they communicate with each other, how they send information, it's just gorgeous. Yeah, so Audrey asked what it's called. I'll type it in the chat so it's bigger. This is called the Connectome. It's the way that the parts of the brain connect to each other. So this is relatively new. The technology that we have is relatively new, allowing us to do this. But you can see really clearly. So check out that corpus callosum right there. That's just gorgeous. All of those connections from one side to another. We can see a lot of these little tracks that are traveling from different places, different neurons of the brain going out to other areas. Yeah, so probably some projection tracks going down there. There's some more of those commissural tracks. You can see a little bit of association tracks going on there. So if you want to look at some beautiful pictures of the inside of the brain, the connectome, I thought that was amazing. And like I mentioned, they're doing studies that show your connectome matches with your personality type. So I don't know nearly enough about that yet, but that's on my summer to learn list, the way that your connectome is shaped by your personality. So there's Dr. Ollis's nerd moment for us here. All right, I see a question about action potentials. So let's talk about that. That would be a great thing for us to review. Yeah, Fanshin mentioning that it is really cool that it's so individualized. I've told you all my husband is a therapist. So the one type of personality test that's actually like supported by scientific research is called the Big Five. So there's 5 traits that you get a scale on, and that's what they compared these connectomes to. Was those Big 5 personality traits. So it's like conscientiousness, agreeableness, like introvert, extrovert. They were able to find some patterns across those in the connectome. So very interesting stuff out there. All right. You have this picture both in lesson number 9 and in lesson number 10. So I don't care which packet you look at. I know Tian mentioned wanting to talk about summation. So maybe look at number 10. If we're in the lesson number 10 packet, where is action potential in lesson number 10? What page is this on in that packet? Yeah, Kaylin, I'm not sure that my husband has actually seen any of my students. I hope he doesn't, because he works in addiction treatment. So I hope that none of my students end up seeing my husband. Although when we lived in Tulsa, he did actually have 1 or 2 of my students, because he worked kind of in outpatient mental health. So he did see some of my students in the past, ironically. Okay, so we're somewhere around pages 9 or 11. We're talking action potentials here. So let's remind ourselves, when we talk about action potential and action potential, remember that how I want you to think about what that word means is a change in charge that moves. Okay, perfect. Yeah, so if you are in your lesson 9 packet, we're page 9. If you're in the lesson 10 packet, page 11. Perfect. It's the same action potential that moves a charge across the membrane of a muscle cell. So the way that I move a signal down the T-tubule is exactly the same as the way I move a signal down the axon. They're literally the same as each other. So the way that an action potential works is we start with a signal. So I always get a signal from one neuron. So here we'll do a little neuron drawing because it's been a while. Okay. We always start with one neuron. That neuron is going to be talking either to another neuron or to a muscle. So we've got a membrane over here, whether it's an axon, or excuse me, whether it's the dendrites or whether it's the muscle. I'll put muscle over here. We've got a place where that neuron is talking to the other cell. The neuron spits out neurotransmitters. It spits out acetylcholine if it's a neuron speaking to a muscle cell. That acetylcholine, we reviewed this on Friday, acetylcholine is going to open up what kind of channels on the membrane of a muscle cell? When I spit out acetylcholine, what kind of channels does acetylcholine open? Yeah, exactly. Acetylcholine is a chemical. So acetylcholine is going to open up chemically gated. specifically chemically gated sodium channels. So before we get to the action potential, and this is before we get to the action potential in neurons too, for that matter, before we can make a change in our membrane charge that's moving, we always start with opening up chemical gates. So acetylcholine leaves a neuron and it goes and either attaches to a chemically gated channel on a muscle membrane or a chemically gated channel on a dendrite. We always start the process of signaling with chemically gated channels. Those chemically gated channels let in sodium. When sodium comes inside, here's my favorite, right? Hey, I just literally bought bananas at the store today. We've got our salty banana, sodium plus, chloride minus. I'm going to steal your thunder because it's been a while since I made my own salty banana. Just going to be real here. So here's Dr. Wallace's salty banana. When I open up a channel, so ACH came and attached to a chemically gated sodium channel. Sodium rushes inside. When sodium rushes inside, my membrane charge gets more, what? Sodium rushing inside. Yeah, exactly. I'm going to get more positive. So sodium rushes in, I get more positive. When my membrane gets more positive, that's going to cause a new kind of channels to open. Who opens when the charge gets more positive? What kind of channel opens when my charge is wrong? Yeah, exactly. Now we're talking about voltage-gated sodium channels. So my acetylcholine opened up the chemically gated sodium channels. Change in charge opens up my voltage-gated sodium channels. So the charge change that happens because of this leads me to open up voltage-gated sodium channels. Voltage-gated sodium channels are found all over, when we're talking about a muscle cell here, they're found all over the sarcolemma. There's a muscle word for us there. Sarcolemma, that's the plasma membrane of a muscle cell. We also find voltage-gated channels all over the T tubules. Remember that those were the tubes that go inside a muscle cell to help the entire muscle cell contract at the same time. And when we're talking about neurons, we find voltage-gated channels on the axon hillock, on the axon, and on the axon terminal. Move those. So when you're thinking about the types of channels places, I guess I'll label this here, voltage-gated channels, here's our list of places where those are found. They're found on the sarcolemma, they're found on the plasma membrane, and as we move a signal across the plasma membrane, we bring it to those T tubules, to those things that go inside the muscle cell. That's going to allow us to contract. If I'm talking about a neuron, my voltage-gated channels start on the axon hillock, where I do summation, where I add my messages together. And if I get high enough, it's going to spread down the axon, down to the axon terminal, where I can spit out those messages. So this that we're seeing here, this picture, showing us the action potential moving in blue, this is showing us what it looks like when a signal goes across the sarcolemma and down the T tubules, or when it goes across the axon hillock, down the axon terminal. Here's how it works. So let's say we did this chemically gated thing. Inside the cell got a little bit positive. It got positive enough for voltage-gated sodium channels to open. Here we are, voltage-gated sodium channels. It was kind of positive right here, so I get positive too. I open up my voltage-gated channels, in comes some sodium. Now the inside, you can see these little pluses here. Now the inside of this membrane is positive. Well, my voltage-gated sodium channels who live next door, right here, they say, oh, I guess we're opening up, because that's what my neighbor did. So now my voltage-gated sodium channels right here open up. And in comes the sodium, and inside gets positive. And my voltage-gated sodium channels that live right here say, oh, we're doing the positive thing. They open up, and in comes sodium. All across the membrane, so all across the sarcolemma of a muscle fiber, all the way down those T-tubules, voltage-gated sodium channels open up everywhere. When one opens up, the next is going to open up, which will cause the next to open up, and down the axon we go. But remember, my cells don't like to be positive on the inside. That's not where they like to be. Here, this would be a good reminder for us. When we talk about the resting membrane potential, the normal charge on these cells, when I talk about a muscle cell, what is my resting membrane potential on a muscle cell? Yeah, there we go. My resting membrane potential on a muscle cell is -90 millivolts. When I talk about the resting membrane potential on a neuron, what's the resting membrane potential on a neuron? Yeah, exactly. A neuron is -70. Hey, make sure we know these numbers. We must know these numbers here. This is normal. If you're a muscle, inside is normally -90 millivolts. If you're a neuron, the inside is normally -70. So when this part of my membrane goes positive, when it goes all the way to +30, this part of the membrane is like, oh, we are mixed up. Because the charge is too high, because it's a charge issue, we're going to use voltage-gated channels again. This time it's voltage-gated potassium channels. Voltage-gated potassium channels allow potassium to leave. Potassium leaves my cell, which makes the membrane charge go back to negative. If we're talking about neurons, we go a little too negative before we get back to normal. Muscle cells don't do that, they just get back to normal. But for everywhere that those voltage-gated sodium channels open up, as the neighbor's voltage-gated sodium channels open up, voltage-gated potassium channels will open up behind it, because their job is to repolarize the membrane to get it back to normal. So when this signal moves next door, now this part of the membrane is depolarizing. My voltage-gated potassium channels, again, will open up to get us back to normal. So all across the membrane, we start with opening voltage-gated sodium channels. We follow that up with opening voltage-gated potassium channels. The goal to be depolarizing the membrane so that we can keep sending a message, but then repolarizing it to get back to normal. What questions do we still have about action potentials and the channels and all that jazz? Are we thumbs up then, since I don't have any questions? Here comes a penguin. I feel like something that I should have started way back at the beginning of the semester is I should have just called it like March of the Penguins. And like that would have been a time that we all just put penguins into the chat. Have a March of the Penguins. Yeah, so Tammy Tay was asking about the Crossbridge Cycle. I have a request for summation, and then we'll do the Crossbridge Cycle, definitely. Okay, let me pull up our picture about summation really fast. Let's use this one. So this is lesson 10 packet. What page do we have this picture? This graph that shows us how summation works. Okay, it might be page 9. Okay, perfect. All right, so this graph shows us how the process of summation works. Let me change colors here. Remember that summation is when a neuron uses more than one message to figure out whether or not it should be talking to its neighbor. So in summation, we add together lots of messages to figure out whether or not we should respond. Some of those messages generate what are called Epsps, excitatory postsynaptic potentials. These messages, when I get them, I open up sodium channels to make my membrane charge get more positive. So anytime a friend, so one of my neighboring neurons here, is sending a message, a chemical that opens up a chemically gated sodium channel, when sodium comes in, that makes the inside of my membrane a little bit more positive. So I got an excitatory message here, I got more positive. I got another excitatory message here, I got more positive. But then I got my first inhibitory message. See how my membrane charge went down? When my membrane charge goes down, I call that an IPSP. It was an inhibitory neurotransmitter. Remember that there's two kinds of ion channels that can lead to inhibitory postsynaptic potentials. If I open up a chloride channel, chloride with all its negative is going to drag my membrane charge down. Also, if I open up a potassium channel, that's going to drag my membrane charge down because all the positives leave my cell. In the process of summation, I don't just listen to this message. I don't just listen to this one. I also listen to this one too. And as I'm listening to all three of these messages, none of these messages were enough to get me up to threshold. Remember, this was another number that we had to make sure that we know. Threshold, -55. When I add together, it's literally math. When I add together the changes in my charge, at the end of those three messages, I didn't get higher than -55. So I don't get to talk to my neighbor. I got another message that opened up a sodium channel again. In comes some sodium. I'm getting closer to threshold again. But I got another message that was an inhibitory message that maybe let in some chloride, and I'm back to being negative again. But then I got this positive message. It opened up those chemically gated sodium channels, and I got all the way to threshold. Remind me again, which kind of channels open at threshold? What kind of channels am I trying to get open right here? Yeah, exactly. It's still sodium channels. I'm still getting more positive, but -55 is where those voltage gated sodium channels open. So the magic number I'm shooting for when we talk about summation is -55. If I can get to this voltage, I will open up my voltage gated channels, and that will allow me to go through this whole process, allow me to talk to my neighbors. So keep in mind, summation, it's all about -55. If you send me a message that makes my membrane more positive, I call that an EPSP. If you send me a message and it makes my membrane more negative, I call that an IPSP, an inhibitory postsynaptic potential. There are two kinds of summation, or two different ways that neurons can receive messages. One of my types of summation, let's go back one more. One of my types of summation is called temporal summation. Temporal summation. The thing for you to keep in mind with temporal summation is one neuron is sending me messages over and over again. So over a period of time, one neuron talks to me a lot. If you compare that to spatial summation, I receive a lot of messages across different spaces. So literally different parts, different dendrites on my neuron receive messages. Spatial summation, more than one neuron is talking to me. Temporal summation, one neuron is talking to me. So it's a difference in the number of neurons that talk. In temporal summation, one person talking to me a lot. Spatial summation, more than one talking to me. That's where we had that analogy in your guided lesson and in your notes about the two ways that you could end up with 99 messages on your phone at once. It could be that you have one person, like Captain Marvel, who just keeps repeatedly texting you over and over and over again. If I've got one person sending me a ton of messages, what kind of summation is that? If it's one person sending a ton of messages, what kind of summation are we talking about here? Yeah, exactly. That's my temporal summation. Remember, temporal summation is one neuron talking, and they're sending lots of messages over time. So temporal means time. So over time, one neuron talks to you a whole bunch. It's like if one person sent you 99 messages. More likely, if you've got 99 messages, lots of different people are texting you. So if I am receiving messages from more than one person, like I can see in this group chat here, more than one person or more than one neuron talking to me, now we're talking about spatial summation. Spatial summation. And that's when multiple neurons are talking. Multiple neurons are talking. So if more than one person or more than one neuron are sending messages, that's spatial summation. If only one neuron is sending a lot of messages over some period of time, that's temporal summation. We mentioned this kind of briefly, but I want to emphasize it since it is on the homework assignment and it's an important difference to know. When we talk about temporal summation, where just one neuron is talking to you, neurons can only ever talk one language. So what I mean by that is, we'll go back to the slide here. This neuron in my picture is shown, it's sending two green messages, two excitatory messages. The only language that this neuron speaks is excitatory messages. At the same time, there's going to be other neurons, different neurons, that the only language they speak is inhibitory messages. Neurons can only ever speak one language. Either they're always going to be speaking positive, or they're always going to be speaking negative. So we have the picture here that shows you, see how this with this spatial summation up here, this neuron always sends positive messages. This neuron always sends positive messages. These neurons down here, one of them is sending a positive message, one of them is sending a negative message. The way that it works in your mind, if we're trying to get you to not do something. So for example, if you ever study the way that tetanus works, which when you step on a rusty nail, how you can have muscle spasms. The problem with tetanus is not that we tell our excitatory neurons to talk more. It's actually what happens is we deactivate the neurons that their only job in life was to send inhibitory messages. So in your mind, in your body, we have neurons that the only thing they ever do is they regulate things. They only ever send negative messages. We also have a lot of neurons that only ever send positive messages. So when we think about something like spatial summation, the way that a neuron listens to all kinds of different messages, spatial summation is the only way that I can get some positives and some negatives, because I need some neurons that always talk negative, and I need some neurons that always talk positive. If one of these friends is the only neuron talking to me, I'm either going to have a whole lot of negativity in my life, or I'm going to have a whole lot of positivity. In temporal summation, it's always either positive or it's always negative. Yeah, exactly. Like Temi Teo put in the chat, neurons are not bilingual. They don't say yes sometimes and no other times. They speak one language. They are always yes or they are always no. So if you are doing temporal summation, we're getting yelled yes all the time, or we're getting yelled no all the time. Temporal summation, it's the same message over and over and over again. Spatial summation is a little bit more accurate, right, to real life. It's a mix of yeses and nos because we have more than one type of neuron talking to us. Yeah, Kaylin said, thankfully, we're not like those neurons where we only say yes or we only say no. Yeah, I agree. Audrey said her daughter says that she's always a no. Her daughter thinks she's always a no person. Yeah, I bet if I asked my 4-year-old, she'd say the same thing. They just made a movie about that, right? The Yes Day or whatever. I have not seen it. About a day where parents just say yes all day long. I don't know if it's any good, so do not say that Dr. Ollis recommended it because I haven't seen it yet. I have no spare time to watch such movies. Okay, Krista says it's good. It looked cute. So if you want to find out what happens when an inhibitory neuron, aka a parent, decides to no longer be an inhibitory neuron and be an excitatory neuron, you can watch the Yes Day in all your spare time, right? Because it's not like you're studying for a test or anything. Does that clear up summation? I think it was Tian who had asked about that. Okay, perfect. Remind me, what was the other topic we wanted to talk about? Crossbridge cycle, right? Was that the last one? Let me go pull that up. Yeah, Crossbridge cycle, perfect. Let's see. So now we're going to go back to the lesson number 9 packet. What page are we on in lesson number 9? Where do we have that cross-bridge cycle? While we're searching, Kaylin asked a good question in the chat. She asked about the specific kind of ion channels that would open when we end up with an Epsp. Can anyone help me out in the chat? If we have neurotransmitters that attach to a channel and cause an Epsp, yeah, Kier's right. So there's kind of 2 parts to that question. The first part to the question talks about a channel that opens with neurotransmitters. When we open with neurotransmitters, we're definitely chemically gated because neurotransmitters are chemical. When we talk about making an Epsp, we need our membrane charge to go up. So the only ion I can move that makes my membrane charge go up is sodium. So when it asks what kind of channels would open when a neurotransmitter attaches to them, and it would lead to an Epsp, that's going to be a chemically gated channel. When we talk about the kind of channel that depolarizes the membrane or helps us to move an action potential, that's when we get to start talking about those voltage gated channels, when we're talking about the action potential, or when we're talking about threshold and summation. But anytime we're talking about neurotransmitters, it's going to be those chemically gated channels. Neurotransmitters are those chemical keys. All right, I heard we are on page 6 of lesson number 9, where we talk about the steps of the Crossbridge cycle. I want to put a quick plug in for looking over the group work that we did in lesson number 9, part 2, that particular video, because if you remember, we made some flow charts that outlined this process. So if you want to go back and try to rebuild those flowcharts to help you study the Crossbridge cycle, that would be lesson number 9, part 2, that video. When we talk about the Crossbridge cycle, remember that for all of this to happen, for us to do the process of muscle contraction, we first had to release calcium. So this calcium kind of shows us where excitation and contraction come together in excitation, contraction, coupling, because at the end of that action potential moving down the T tubule, it caused some proteins to press on our mechanically gated calcium channels. They spit out calcium. Calcium goes down to those push pin proteins, troponin, and causes the push pin to pop off, which makes that spaghetti noodle, tropomyosin, makes that slide out of place as well. That allows the Crossbridge cycle to start. So even before any of this stuff happens, remember that we had to spit out that calcium and we had to move away those regulatory proteins. Step 1 of the Crossbridge cycle is very straightforward: Crossbridge formation. In Crossbridge formation, myosin attaches to actin. Myosin will always attach to actin as long as myosin is in the upright position, and as long as those myosin binding sites, these little circles we see here, as long as those are available. So anytime we spit out calcium and we move away tropomyosin, anytime these little binding sites are available and myosin is in the right shape, we will form a crossbridge 100% of the time. Crossbridge formation. Myosin attaches to actin. Step 1. Step 2, the power stroke. When you're thinking about the power stroke, this is the stage where you actually make muscle power. This is where movement actually happens. The reason that movement happens is because now that myosin is attached to actin, it's like, I don't need this garbage on my back. I'm gonna let go. So it spits out this leftover stuff, which makes it change its shape because it no longer has that pressing it forward. So myosin bends over while it's still attached to actin and causes actin to slide, the power stroke. But this is where, remember, we were talking about rigor mortis. This is where we get stuck unless a new molecule of ATP comes along. See, myosin really wants to be holding something. So if it doesn't have ATP to hold on to, it's like, I'm just going to hold on to actin forever. Like, this is my jam. We're just going to chill out together. If ATP comes along, ATP is a shinier, nicer toy than actin is. So myosin will attach to ATP if it's available, which will cause it to let go of actin. This is that step called crossbridge detachment. Remember, this is the step we can't do in rigor mortis because there's no ATP floating around. If ATP comes around, myosin attaches to ATP and lets go of actin. It doesn't care about actin anymore because it's got the shiny new toy over here. But once myosin starts holding ATP, it realizes if I break this ATP, I can change my shape. So my 4th step of the Crossbridge cycle, cocking the myosin head, we go from being bent over, holding on to ATP, to standing straight up, holding on to ADP and phosphate. Remember, let's do a little drawing here. ATP is adenosine, an A group, with 3 phosphates, ATP. If I can break up this chemical bond right here, it turns into ADP because it's only got 2 phosphates, and then we got that little phosphate chilling out. So what myosin does is it chops up this little part right here, which gets a phosphate free, which leaves behind ADP. The energy that was right here, I'm using to go back into the cocked or the upright position. And remember, if I'm in this upright position, and if my myosin binding sites are available, I will start over and I'll just keep going and going and going until I run out of ATP and I can't detach anymore, or until there's no calcium floating around anymore and those myosin binding sites are covered. How do we feel about the Crossbridge cycle? Thumbs up, thumbs down, flexing our muscles. Yeah, you're welcome, Tummy Tail. Absolutely. All right. I think I'm going to call that a day. But if there are any last minute questions you want me to answer before I turn off the recording, hit me up in the chat really fast. I will give us our penguin because it's been way too long. Remember, when you are taking that exam, if you have any issues with Proctorio, if it kicks you out of the exam, please contact Proctorio first. I cannot get you back into the exam, so please get in touch with them right away. Try to get that exam finished, and they should be able to get you back in to help you with that. Please plan to try to take it not right at the very end of the window in case something goes wrong. Full disclosure, I will probably not be up until midnight tomorrow night, unless I'm up with a sick baby, but fingers crossed he'll be feeling better tomorrow. So please try to take it earlier in the day tomorrow. Yeah, just like Fanchin said, you all got this. I'm rooting for you. I'm going to have open office hours tomorrow from 1 to 2:30. So if you think of questions as you're studying between now and then, please feel free to stop by. I would be happy to chat with you in that session or via email as well. So here's my penguin saying, you've got this. Best of luck to each of you with that exam. Next time I will see you for class is Wednesday. We will cover this week's lab stuff. So good luck on that exam.